Hello friends, this is Gopal Agrawal from NIPEM Mohali, student of biotechnology, and today we are going to see some of the tips for cracking or for preparing the GATE Life Science. So initially we will start with the pattern of the life science paper. So in the life science paper you have chemistry, that is compulsory for everyone to attend, aptitude, that is compulsory, right? And then there are the two optional subjects to be chosen. So the options that have been available are biochemistry, microbiology, food processing technology, zoology, botany. So you can choose any of these two subjects, right? With chemistry aptitude compulsory, it means you can choose chemistry aptitude, that is compulsory, and from the option you can choose either biochemistry, microbiology, or biochemistry, food processing, or anyway whichever subjects, two subjects you like, you have to choose. Today we will just focus on biochemistry and microbiology. So now let us see first the paper pattern, right? So there are question number 1 to 5 will be of 1 marks each, and they will of total 5 marks, and they will be of aptitude. Then question 6 to 10 will be 2 marks each, right? They will be of 10 marks, right? And it will be again of aptitude. Question number 11 to 15 will be 1 marks each, 5 marks, and they will be of chemistry. 16 to 25 again 2 marks each, so 20 marks, and chemistry. So aptitude will contain 15 marks, chemistry will contain 25 marks. Okay. Now, in any of this, either in 1 mark or 2 marks, there can be questions where there are no option. It will be like fill in the blanks, right? So that sorts of questions are also been asked in the GATE, right? So it's not compulsory that every question has an option in GATE. There are many questions where there are no options been given. They have been just given as a blank, and you have to fill that blank. And they are just generally based on the mathematical calculations, right? Okay. Next is we will just focus today on biochemistry and microbiology. So I have taken over your biochemistry and microbiology. So biochemistry, you are having again 1 to 10 questions will be of 10 marks, and another 11 to 20 question will be of 2 marks each, so it will be of 20 marks, right? In 1 marks there are easy questions, right? But it depends on your preparations whether you feel that questions are easy or not. But mostly in that type, in sort of that sort of questions, there are no question without option, but we can't say it's a gate, right? In 2 marks you are having met the column, right? Mostly 10 match the columns have been always been asked. In 1 marks also sometimes there can be asked questions without without options. The blank that's been given, right? So they are basically simple mathematical calculations here also. In microbiology, again there are 1 to 10 questions of 10 marks, and here also there can be questions without options as given as a blank, right? And 11 to 20 questions are of 2 marks each. They can be here there are the match the columns. So total questions, now you count from for aptitude, chemistry, and the two optional subjects, there are 65 total questions, and 40 marks is for aptitude and chemistry, remaining 30 marks for each subject, that is 30 for biochemistry, 30 for microbiology. So total marks will be of 100. Let us start with the general aptitude, right? So there are question number 1 to 10 will be of general aptitude, right? And it will be carrying of 15 marks, right? With 1 marks and 2 marks questions, right? So now what will be there in your aptitude? So they will generally ask grammar, right? Vocabulary, they give a word, you have to have a synonym or the antonym of the word. In the grammar they give you most of the sentence, and they either ask the some of either the sentences be right, or what is the meaning of the underlined line that has been given in the sentence, right? Logical questions have been there mostly, right? And quantitative aptitudes, right? Quantitative aptitude questions are also been there in the GATE, right? Simple, basically you don't have questions on time and work and all sort, but it considers all the quantitative method, right? They are simple, right? If you go and solve them, they're a simple way, but it needs some practice also, right? So before the exam you need to have just 1 week practice of the general aptitude, right? So if you practice 1 week also, or if you practice 15 days, it's most, right, it's enough, I think so, for the general aptitude, because the general aptitude is not so hard over there, right? As for example, in 2015 paper, they asked what kind of sound a frog makes, right? It's a rose. It's like it was some options were been given over there. So it's that's likes of questions have been asked over there, right? Uh, then which books to refer for the aptitude? You can refer R.S. Agrawal book, a very famous book for the preparing the general aptitude, and you can also visit the website called as the indianbix.com, right? So here also there are some many questions given topic wise, right, on the website. You can visit there and you can see what sorts of questions have been there. Basically these questions are from R.S. Agrawal only. So once you solve R.S. Agrawal, and if you want to have a practice of seeing how much you have prepared, you can visit the website, you can solve the questions, right? The solutions have been given there also. Let's come to the chemistry, another compulsory subject. So the total number of questions asked are 15 and marks are 25. Now there is no basic criteria that at least you should get something, that means out of 25 marks you need to score 15 marks. There is no such criteria in GATE, but the main thing is you should attempt that questions, right? Without attending that questions, it's not they're not going to consider you further. So if you at least get 1 or 2 marks, also that is enough, but you need to attempt the questions, right? There is it's no hard and fast rule that you should get at least minimum this much marks in the chemistry section, this much marks in the aptitude. No such rules. Your total marks have been counted, but there is a need for you to attempt the questions, right? So but you have to score some marks, right? So here there is a syllables of your chemistry section. So it basically consists of atomic structure and periodicity. So now in this we will have highlighted all the things that have been most important, right, for you to prepare for chemistry in the GATE. Mostly the questions have been asked from this highlighted yellow ones, right? So for example, you are having this ionization energy in atomic structure and periodicity. You're having ionization energy, electron affinity, electronegativity, atomic size. From here, from ionization energy, sometimes questions have been there from electronegativity, electron affinity, atomic size. So remaining this, the Planck's quantum theory and all this, you just need to go through, right? You cannot neglect them. You have to just see one or two times, right? There's all these concepts, but you need to focus mainly on this, right? Ionization, electron affinity, electronegativity, atomic size, right? You need to go in the depth, some depth of it. If you see superficially this all Planck's quantum theory and this, that's enough, right? That's enough. But if you see ionization in superficially, that may cause the problem, because sometimes questions have been asked in the depth from this topics in structure and bonding, right? Most important is VSEPR theory, very important. This molecular orbital and VB approaches for diatomic molecules is a very important topics. Every year I think so one question has been asked from here, and it's not that only one question has been asked. Why to prepare one questions are very important, right? It's like a drop of water will fill your sea, right? So you have to grab one marks, right, in order to get scored, right? Another important thing over here is Born-Haber cycle, very important thing, right? Basically this whole topic, right, this structure and bonding is important, right? But yeah, but you need to focus mainly on VSEPR. More importance should be given to VSEPR theory and shapes of molecule. Born-Haber cycle, very important. Born-Haber cycle, VSEPR, molecular orbital and VB approaches, these are also very important things, right? Okay, another thing you can also focus here is on bond length, bond angle, bond energy, right? This sorts of thing will also be important. Hydrogen bonding, in which elements there are hydrogen bonding, right? You should know that, right? In which atoms there can be hydrogen bonding, that is hydrogen, and within the electronegative atom, that is fluorine, oxygen and nitrogen. From there only the hydrogen bonding can occur, in which there is van der Waals interaction. Such type of questions will be asked, right? Then you have sp and d block elements. Here this all sorts of thing, oxides, halides, are not very much asked, right? But yes, this can be asked. The configurations of p and s, right? What are the coordination complexes, right? Coordination complex from here also every year one question has been there. Valence bond theory, crystal field theory, very important thing. From here also questions have been there. Geometric magnetic property, very important thing. Which are diamagnetic, which are paramagnetic, right? These type of questions are also been there in GATE every year mostly. Then from chemical equilibria, right? Qualitative properties of solution, ionic equilibria in solution, this sorts of thing you can just have once a look, right? Electrochemistry, another very important section in chemistry. From here every year 1 or 2 questions have been there, mostly based on the Nernst equation and ENF, right? Colseger-Rolls, right? There are fill in the blanks. From the Nernst equation mainly you will see the fill in the blanks without any options, right? So that's important thing, right? So you need to prepare this thoroughly, right? You do not have to go just superficially. You need to go into depth, right? Go in the depth as much as you can, understand the basic concepts of each and everything, not just mug up. Reaction kinetic, very important. Again, you from first second order equations, there will be given the how to find out the rate constant. Very sorts of things can be asked from the reaction kinetics, right? Thermodynamics, very important again, right? His law, Hess law, heat of formation, internal energy, enthalpy, Kirchhoff law, very important things for the Kramnik section. From here also mostly questions have been asked, right? And from here also there can be chances of asking fill in the blanks, so you need to focus thoroughly on this. Another is Totten rule. From here 2 or 3 times the questions have been asked from Totten rules, right? Then first, second, third law of thermodynamics you all know, but you also need to go through them, right? Then there's the basics of organic reaction mechanisms, right? All this highlighted one. From here one question has always been asked. Dielder reaction, Markovnikov rule. Markovnikov rule is very important. Kharasch rule effect is very important. Addition reaction is very important. Dielder. From this many times questions have been asked. So let us see the structure. The last part of it is structure reactivity correlations. From here optical and geometrical isomerism should be given a basic importance, right? Concept of atomicity also is given importance over here, so you can prepare this. Now let us come from where to study. You can study Wikipedia. Why I'm suggesting Wikipedia is very many of you must be unfamiliar with the basics of all these topics, right? So in the Wikipedia you can first see the basics of all the topic, and then you should need to go into depth. If you will go directly into depth in some of the chemistry book that you will see, right, you will not be understand directly what are diamagnetic, what are paramagnetic, right? You may not able to understand many of the terms that have been used over there, right? So once in the Wikipedia, if you go all this to this topic, see what are the basic thing, and then you move towards the book, reference book, right? One of the reference book that I will suggest, Atkins Physical Chemistry. From here you can have electrochemistry, right? You can study from your electrochemistry, and qualitative properties are also been given in these books, right? So you can do this. There are many numericals also been given in the Atkins Physical Chemistry. You can go through that also. Google, yes, another thing is Google. After Wikipedia, we suggest to Google. Search on the Googles related to various topics. You will get various PDFs, right? Just download them, just look towards them, right? This is the way of preparing. This is not a just called a smart work, not hard work, right? Because you need to have a basic concept. There's no hard and fast rule that you need to learn from a reference and standard book. You need to be you're much clear in your concepts, right? And your concepts can be clear from the basic things, right? So basically you should first prefer all these 3 books: Wikipedia, Atkins, Google, and then you can move to the another chemistry books also. You can just have a looks over them. Now let us come to the biochemistry. So here the total number of questions are 20, marks is 30, question pattern is 1 marks and 2 marks, right? There can be question with option, without option, as I said, as a blank. Match the columns will be there, right? Attempt the match the column when you are sure. Do not be in a hurry, right? That okay, I'm getting this, I know. See, many times when you are sure of only one option of 4, right, in the matter column, and remaining 3 have been just twisted. It may give you the blunder, it may give you negative marks. So unless and until you are not sure of at least two correct answers in the method column, do not attempt it. When you're sure of 2, you can take a risk, right? Then what books to refer? Larringer. I will just tell you to read Larringer. If you read Larringer thoroughly, at least twice to thrice, believe me, you will get very good marks in biochemistry. But you not have to read it as a novel. You need to be very much thorough in what you read, because once you will see the question, this whatever the syllables of biochemistry, you will find that just been copied and pasted from the Larringer. Same chapters, same topic in Larringer you will get, and believe me, every year question paper is been set from Larringer. Mark my words. If you do Larringer thoroughly, twice to thrice, believe me, you are going to get very good marks in biochemistry. Okay, but you need to be thorough. Your concept should be clear, right? Because in Larringer it's not easy to understand Larringer. At the first time that you will in Larringer, you will not get anything. Believe me, you will just be empty barrel only again. But when you're going to read second time, third time, fourth time, right, you will be very much clear in your concepts, right? And this will be very much useful to you in other examinations also. No need of other books if you read Larringer, or if you want to be much easier, the Styler book can also be referred, but basically you should refer Larringer. Larringer should be the first preference. Okay, now coming to the biochemistry, all these questions, all these chapters are there in your Larringer, right? They've been copied and pasted. If you want to see, you can see them. Okay, so water, yes, water is important. Water chapter, second chapter of the Larringer, very important, right? Then you have amino acids. Yes, amino acid is also important chapter, right? Then biochemical, chromatography, very important. Chromatography, very important. I'm using this twice. From chromatography, size exclusion, ion affinity, they have been given over there. So you just be prepared. What is the basic principle of this chromatography? For example, in size exclusion chromatography, which molecular weights, so these larger molecular weights or smaller molecular weights will come elute first from the column. These types of basic things you require, right? Then you have these enzymes, very important enzymes. As for example, this year it was been asked that what is the mechanism of lysozyme, right? So from these enzymes also one question has been asked, right? Enzyme kinetics. This should be done so perfectly that even if you get early, wake up early in the night, you should be able to answer any of the questions from this. This chapter is so important from the Larringer. You would just see competitive binding, non-competitive binding, uncompetitive binding, right? Graphs of it, what are the various equation, Michael-Menten's equation, right? You need to be thorough in enzyme kinetics. Very important from here also. 1 or 2 questions have been every time been asked. Metabolism and bioenergetics, another very important chapter, right? From here, delta G, delta H, change in the free energy, enthalpy, right? Here also questions have been there. 1 or 2 questions have been there. Now, my suggestion regarding this TCA, glycolysis, and all sorts of things, you should do this at the end, and here mainly focus should be given from where the ATP is generated, how many ATP is generated. You not need to be thorough in the various enzymes which have been there in the various pathways, right? You not to be much thorough in this, means not thorough in the sense that you not to be remember the mechanism of how each and every enzyme. You should just remember which enzyme acts at which step, right, in the cycle, what amount of ATP is utilized or synthesized during that cycle, right? What is the importance of that cycle, and where does this cycle occurs? These are the basic things that you should know of each and every cycles. Okay, Then biological membrane, transport across membrane. This all things also you can find in laryngia, and it's very important thing. Another important things you will see is DNA replication, transcription, translation is also been there in the laryngia, right? Biochemical regulation of gene expression is also been there in laryngia. You can just see to it, right? Lac operon will be there in biochemical regulation of gene expression. That's also important thing. And you have an immune system that is active. In immunity, you can refer either QB because in QB immunology book has been there, right? That's very good book. You can get all these things in that. Or in a laryngeal, there are sort of things of immune system, but in laryngeal immune system is not been given in detail, so you can refer QB, KUBY, QB immunology books, right? So then next comes to the microbiology, right? So again there are 20 questions and 30 marks, 1 marks and 2 marks question. Again there will be question with option, blanks, match the columns, right? And in order to refer for Maringer, Pelzar is the best book. Just sit with Pelzar. All the topics whichever they are given in the microbiology syllabus in the gate are been given in the Pelzar. Okay. After Pelzar, you can for human genetics, it will be a part of the microbiology. You can refer any of this, any one book of this, either Prescott, right, or you are having Goldstein, Levin's gene, molecular biotechnology. From any three, any one. Prescott is nice. Prescott, you can refer it. Prescott is given, and Prescott language is much easier. You can just understand in Prescott. No doubt in these two books also language is easier. Any one of the books from this three you can refer. There is no hard and fast rule in there, right? But Pelza you need to study once. Okay, coming to the syllabus of microbiology. So there is a discovery of microbiological world, landmark discovery, very important from your which scientist discovered what, when was this discovered. Every time questions are in there, so you cannot neglect this, right? So landmark discovery are very important things, right? Then you can have a role of microorganism is also important. Methods in microbiology, very important. The whole topic of this is very important. From electron phase microscope, contrast microscopy questions have been there, right? Then you have microbiology, taxonomy and diversity. This is also very important, but it's not need to be much thorough in this of the classification. Classification is not mostly asked, but you need to be thorough in molecular approaches to the microbiological taxonomy. You can get in the Pelzar. Then the very important thing is this prokaryotic cells, cell walls, cell membranes, right? These are the very important things from your questions have been there in the case. Then this microbiological growth, you see it has been highlighted twice. This is very important thing. Now for this you can have another book, I think so it has been given in the Pelzar also, microbial growth. But if you want to go see in much details, there are some biotechnologies book, right, that you can refer for the microbial growth. But in the Pelzar also the topic has been given, right? Then you have a control of microorganism. Here the evaluation of the effectiveness of the microbial agent, that is Friedel-Walker coefficient, right? All sorts of things will be there here, so that you need to be thorough in that. Microbiological metabolism is also important thing, right? Here the will repeat the citric acid cycle, pentose phosphate pathway, as you will see in the biochemistry, right? So the other things will refer. Microbiological disease and host pathogen interaction. From here again humoral and semitic immunity. Vaccines have been important for which kind of disease, which sorts of vaccines, toxoids, these sorts of things have been utilized. You need to know that. Chemotherapy is important, right? Then microbial genetics, very important in the gate. From your 2 or 3 questions or 1 or 2 questions are always been asked, either in the method column, fill in the blank, or question with options, right? So these sorts of things can be asked, and for this I told you either you can refer Prescott or Levin's gene, any of the book, whichever you like, you can refer here. Now comes when and how to prepare. So you should start at least 6 to 8 months before your exam. Prepare proper separate notes for individual topic. Go into the depth of individual topic. Do not only have a superficial view, understand the basics of each and every topic. Do not cramp it, right? Revise each topic twice to thrice before moving to the next, right? You should not be in a hurry that today Lamon let us complete 10 topics. If you complete in each day 2 topics but with thoroughness, it's enough, believe me. You have to solve MCQ related to one question and then to move to the next topic. Once you read one topic, understand the basic concepts, either see solve some MCQs, right, that have been there. You can get various MCQs of the life science, right, solve the MCQ particular to that topic, and then you move to the next topic. So this will help you gain confidence. Secondly, your concept will be again much clearer. Third, you will be able to remember it. So there are 3 benefits of it. Never be in a hurry to complete your syllables, right? Be thorough in what you learn, right? Do not cramp, do not be in a hurry, right, in order to complete your syllables, right? Discuss topic among your friends, ask them question related topics. So if you have another friend who is in life science, just ask your topic question related to him. So this will again help you in clearing a concept, because if he does not know anything, right, you will explain to him, so your concept will again be much clearer. Secondly, you will able to remember it, right? Your topic will be much more clearer and will be fit in your mind. Solve their queries if they have any query. 1 week before exam, be relaxed, stay calm. Do not be in a tension that, oh, there's a GATE exam. Be relaxed, right? 1 day before exam, just revise the notes that you have made. No new things you can you need to learn. If you read new things, but you can, but first you finish whatever you have made the notes. During the exam, be calm, relax, and enjoy the exam. Very important thing, you should enjoy the exam. You should not be that, oh my god, I'm giving GATE. Nothing sort of things. You should enjoy your exam. Come on, it's a GATE. You should come inside. Okay, I'm giving GATE. You should be proud of it, right? Cracking tips. I cannot say cracking tips. It just will help you to. There's no that rule that if you will follow this will crack, but it just sort of help you to crack the paper. So you just first attempt biochemistry in my view, then microbiology, then aptitude, and then chemistry. Biochemistry first, because there may be some sort of thing that you remember easily in biochemistry, and the questions in biochemistry are very easy, right, once you get a thorough view of the laryngia, right? Then microbiology questions are also very easy. Then you have aptitude, and finally chemistry. Mark only those answers you are sure of. Do not bluff. Most important thing, be sure of what you are marking, right? If you see that answer is A, be sure that answer that you are marking is A only, right? It's not B or C, because sometime in hurry you just make the question paper, you just mark wrong options, right? Another thing, in the GATE on the online system, there is an option to see the whole question paper. So in my view, you just open the whole question paper section, because one question when it will come, right, you do not mark one answer, right, one question, then another question also you don't know. So in the third question will be nervous that, oh no, I don't know this question. But once you see all the questions, right, in the option that even been on the bottom side of the screen, once you see all the questions from at least 25 questions, you know 15 questions, then you have a confidence, yes, I know something. But in a one question you don't know, second question you don't know, third then you'll be nervous, fourth if you will know, then you will mark something wrong. So it's better that you see the whole question paper first, right? And there is an option of marking the question answers in that also, right? So you can see the whole question paper and you can mark the questions in that also. In match the column, if you are if of 4 you are sure of 2 or 3, then you mark the answer. If out of 4 option, if you are sure of 2 also, you can take the risk of marking it, right? Attempt the question for which there are no option, they don't have negative marks. Yes, there is a most important benefit, I could say, for the question in which there are no option and they are simple mathematical questions only. You just need to know the formulas over there, right? And if you know that, believe me, if you get any of the answer by placing it in the formula, you should write there, right, they don't have negative marks. If it is correct by chance, you will get 2 marks or 1 marks as per the question. Aptitude answers are logical and easy. Do not indulge yourself in a hard and lengthy calculation. Yes, some of them they think over only. In aptitude, do not think much. They are much easier, right? Don't go in a very long calculation of 2 to 3 pages and then derive an answer that is wrong. Simply the calculations are easy only, right? Whatever you learn, be thorough to it and get your basic concepts clear for each and every topic, as I've been saying from back. Okay, one more help that I can make you is there are YouTube's video for the life science pertaining to the Lehringer biochemistry. So you can get it from the YouTube, and it have been made from myself only. So in these videos you will focus on the each and every topic individually of the life science, right, with a detailed explanation. And presently you will get in the YouTube sequencing of polypeptide, association and dissociation, Hill's equation, enzymes, various topics, right, individual topics on the life science Lehringer only. So from there you can have a detailed explanation of each and every topic in the videos. And your suggestions are always welcome at the given ID gopes978@gmail.com. Please share this and other videos. Thank you and meet you soon. Bye.